This set of slides asks the question, if all of the genomes are transcribed, well, what does all of the RNA do? And attempts to answer those questions. The Human ENCODE project, which was sponsored by the NIH, selected a spectrum of genomic features and then analyzed these features in great detail. But it came to the surprising conclusion that most of the genome in both directions transcribes at some level. The central dogma, of course, is that the DNA stores the information, the RNA uses the information, and the proteins act. So it's the role of the RNAs that's most interesting to us. Genomic programming is such that we go from 1 cell to 10 trillion human cells and 100 trillion, including the bacteria. But at cell division, how does each cell regain its nuclear structure and its differentiated state? So that it can control the static and temporal behavior of the genes and the non-coding elements. Well, what does the rest of the DNA and RNA do? The genes comprise only 2 to 5%, and the pseudogenes, the non-coding elements, comprise 40% of the genome in the Riken transcriptome that I have. Well, what do the non-coding elements do? And I've tried to understand what that is. But 50% of the mammalian genomes characteristically are these highly repeated sequences. And so what do the highly repeated sequences do? And what should we be looking for? And my sense is that we should be looking for an RNA-based mechanism that could use all of the information in the genome to orchestrate the many different behaviors of cells. So in the last decade or so, we've had a tendency to recognize that the highly repeated sequences have a tendency to cause problems in almost any genome analysis program. Well, the usual response has been to mask out these sequences, and the following is a sample random intron. You can see the lowercase sequences. But when you mask it, of course, there's these ends say we're not going to consider these when we do searching. But of course, these ends are part of the intron and have been transcribed and therefore present. And the question is, well, what do they do? So the central insight of this talk is that until I looked at the masked and unmasked intron sequences, I never considered the question of how the connectron sequences, the sources, the RNAs, are freed from the transcript. I realized that 50% of any genomic sequence, including the introns, are these highly repeated sequences, and that the highly repeated sequences, if they're put into the introns in both directions, can form reverse complemented pairs that are at least 20 bases long, and that Dicer could recursively act on these to clip up the transcript into a collection of small fragments. This is the central insight. Well, introns get excised either by self-excision or by spliceosomes, and so this shows the self-excision. But of course, the ends of the intron have to be close to each other in order for this to happen, which means that the intron has to be folded in some sense as it's just been synthesized. And this is the spliceosome, which is a very complicated machine that's structure of which is being determined. But that just shows that there are two classes, the simple class and the more complicated class of intron, and that nature deals with these both in turn. So the introns get excised as the transcript develops, and by one of the two mechanisms. The introns then are a separate molecule free to move about in the nucleus. Well, Dicer is an enzyme that can clip any double-stranded RNA sequence that's 20 base pairs to 2 turns, long or longer. And these highly repeated sequences in the introns dominate, I think, the folding of the introns. So when Dicer acts, it cuts the molecule, the intron, into 3 separate molecules, 5' to the Dicer site, the loop, and then 3' of that. So this shows how a single molecule, an intron, when a dicer stem emerges, can only just be transiently enough for dicer to recognize it. It clips the molecule at the dicer site, and then you have the loop, and then the 5' and the 3'. And then, of course, each one of these can be decomposed. So you have a whole cascade of events. And this dicering, recursive dicering, goes on until there are no more reverse complemented strand pairs. This page shows the result of an algorithm that I wrote that starts with a 20,000 base intron, where there are 7 different dicer clipping sites, and then it winds up with 76 distinct fragments out of the 306 fragments that it found. Well, the GWASs came along a couple of years ago, and when they first occurred, people thought that the results were quite stunning, and so lots of people went into determining genome-wide association studies. But somehow in the last years, these studies have not been as fruitful as they might have been, or at least people thought they might be. So one question is, what's the role of the SNPs in intron refolding? Well, since the gene coding and non-coding elements are a very small portion of any eukaryotic genome, the SNPs will affect these in only a small way, and this is what happened in the first GWAS studies. The SNPs affected the exons, and that changed the proteins, and that caused the disease. But many of the SNPs are not in genes, because the genes are just and the non-coding elements are just a small portion of the genome. But my sense is that because the highly repeated sequences are 50% of the sequence of most mammalian genomes, that the SNPs are going to have a huge effect on the highly repeated sequences. So what SNPs mainly seem to do is that they can affect the dicer cascade. And if at the bottom, if the pathways of dicer decomposition of various introns are affected, well, then the collection of fragments that you have as a result of the dicer cascade could be quite different. And I'm in the process of showing that now. The SNPs may have a profound effect on reverse transcription, since that's the largest portion of the genome. And then, of course, we haven't got the foggiest idea of what the role of reverse transcription plays in cellular behavior and differentiation. So the gene coding elements and non-coding elements produce the RNA. The editing excises the introns. The connectron functions. If the connectrons are to function, then they have to be relatively small fragments that can act locally. Dicer acts to decompose introns into collections of small fragments. The dicer-induced clipping cascade frees the connectron sequences so they can act. And since the HRSs, the highly repeated sequences, constitute 50% of eukaryotic genomes, in principle, 50% of the SNPs will be in these sequences. So there should be a strong connection between the highly repeated sequences in the introns of genes and non-coding events and non-coding elements, and even in these reverse transcription cases, which we know very little about. So the SNPs may change the dicer cascade, clipping cascades in a permanent way. And we don't know anything very much, but reverse transcription may be very important in cellular differentiation and development.